I can't help but comment on that last issue there. It's also another regulatory quagmire. I think of the years it took us to get grass status for canola in the US, and yet people in India and Europe had been eating this stuff for literally thousands of years, but it took us years and years and years to get it approved in the US. I think of a smaller, much more insignificant example, but here in Saskatchewan, we grow a berry called the Saskatoon berry. It's an Amelanchier species, and it's not grown on large commercial acreages, but there is some commercial production. When we tried to export this to Europe, wow, did we ever run into a hornet's nest. Fortunately, there was some record of this. I believe it was in Norway. There was a species of it had been grown there, so we found this loophole and snuck through it. But that's the problem with crops that, introducing new crops. Anyway, you didn't come here to listen to me, you came here to listen to the speakers. Well, as I indicated, we were dealing with both the quantity and the quality side, so now we're going to hear about some aspects of the quantity side. Our next speaker is Steve Long. He's the Gutgesell Endowed Chair in the Department of Plant Biology and Crop Sciences at the University of Illinois in Urbana. Steve Long was elected as a Fellow of the Royal Society in 2013. He is a Fellow of both the American Academy for the Advancement of Sciences and the American Society for Plant Biology. Steve has published over 250 peer-reviewed journal articles, including original published research in Nature and Science. He's another one of our speakers that tells you the quality that we've had throughout this, who is listed by Thomson Reuters as one of the most influential scientific minds in 2014, and one of 180 most highly cited authors in animal and plant biology in 2014 and 15, and one of the 20 most cited on global climate change. He was recognized in 2012 with the Marsh Award for Climate Change Research from the British Ecological Society, and also the 2012 Kettering Award from the American Society for Plant Biologists. And in 2013, the Innovation Award of the International Society for Photosynthesis Research. And that is real area of expertise. This is Mr. Photosynthesis. So please help me welcome Steve Long.
First, my thanks to Morris for inviting me to participate in this meeting. I've had a great time until now. So I want to, it's a rather provocative title. I want to tell you why I think photosynthesis is the final frontier in improvement of crop yield potential. And by crop yield potential, I mean the genetic potential of a plant to yield given optimum conditions at a location. So this is not to say that disease resistance, pest resistance, better agronomy, etc., isn't important. Of course it is. But if we look at the Green Revolution, most analyses suggest at least 50% of the yield increase came from improved genetics, and much of the remainder came from agronomy that supported that genetics to achieve that yield potential. So this is a picture of, I was lucky enough last year to meet with Longping Yuan, who is a winner of the World Food Prize and has been breeding rice for over 50 years. And there's one of his cultivars behind us. And the point he's making to me is that his current cultivars are not managing to fill all of their caryopses. And he's saying, you physiologists need to tell us markers, means to get more source to fill what we're breeding. And so that's what I want to try and tell you about. So this is where I work at the University of Illinois, the Carl Woese Institute for Genomic Biology. But we're not detached from reality. Right in front of the institute, in the foreground, are the Morrow plots. And this is the longest running experiment on sustainability outside of the Broadbalk experiment at Rothamsted. So since 1876, corn has been grown every year on these plots with a range of treatments and also one set of plots with no treatment at all. That's now kind of yielding about half of what it yielded in 1876 if you use the similar material on there. But there are also other areas where the yield has remained stable. And my second home is University of Lancaster, Lancaster Environment Centre. So if we look at sort of what we've achieved with probably the two most important direct calorie sources for humans, rice and wheat, we've really achieved a steady increase in those yields over the last 50 years. And so if we project forward, we might say average yields of rice per hectare will be over 6 tons per hectare by 2050. However, according to projections that have been made by the United Nations Food and Agricultural Organization and more recently by David Tillman's team at the University of Minnesota, what we will need by 2050 follows these dotted lines. So even if we can manage to keep on improving yields per hectare as we have in the past, we'll fall far short of possible projected demand. Now, of course, many factors could affect this demand. We could move to an entirely vegetarian diet, there might be less wastage and so on. But we're taking a risk if we don't try and do something, if we don't insure against this possibility. And we also have to look at the fact that actually yields of these crops, if we look at rice, wheat, soybean, are stagnating. The largest producers of rice are China, India, and Indonesia. In the 70s and 80s, those countries were improving yield per hectare by about 30% per decade. If we fast forward now to the first decade of this century, we're into single figures. Yet in the 2000s, China was investing 100 times more than it was in the 80s in improving rice yields, which is telling us it's getting harder and harder to get less and less. And what we might be looking at is a pattern more like this, where if that stagnation continues, we will fall a long way short of projected demand. So why might this stagnation be occurring? Well, if we look at what determines yield potential, i.e. what a crop can achieve at a given location, giving optimum conditions, and this is an equation that was developed by John Monteith at Rothamsted in the 70s, then we can say the yield, tons per hectare, that we harvest is equal to the amount of solar energy available over the growing season, multiplied by how good the crop is at capturing that solar radiation, how good it is at then converting that captured solar radiation into biomass, and how good it is then at partitioning that biomass into the part of the crop we care about, i.e. the grain of wheat or rice, the seed of soybean. So if we look at a modern soybean cultivar growing on our university farm, and we measure these different efficiencies, what we see is that the interception efficiency of photosynthetically active radiation is pretty high, approaching 90%, which is quite remarkable given that the crop has to close its canopy before some of that radiation is not falling on bare soil. If we look at partitioning efficiency or harvest index, that's 60%. So if we're going to have some pod, some stem remaining at the end of the growing season to bear that seed, 60% appears pretty high. The conversion efficiency, however, looks very low, of visible radiation just around 3%. Now, if that is a little bit misleading, because if we look at the theoretical conversion efficiency, if we go through all the steps of intercepting visible light through to making carbohydrate, the theoretical efficiency is around about 10%. So we are far short of that with 3%. But of course, 10% is much lower than what we could expect from other efficiencies. The interception efficiency, however, is almost as it's theoretical, and the partitioning efficiency is not far away from that. And indeed, partitioning efficiency is something that was improved hugely in these crops during the Green Revolution. It more or less doubled during the Green Revolution. But we're now running out of further opportunity to improve that. If we look at conversion efficiency, however, the Green Revolution appears to have done almost nothing for that. Pre- and post-Green Revolution cultivars show much the same conversion efficiency. So this, of course, raises a long-running question. Well, conversion efficiency, of course, is photosynthesis primarily. And there are a lot of arguments, particularly in the 70s and 80s, that improving photosynthesis wouldn't do anything for crop yields. But in the 90s, people became concerned about rising CO2. And so a lot of experiments were conducted on how will crops perform under elevated CO2. CO2 is a limiting substrate for photosynthesis in our C3 crops. So if we boost it, do we see an increase in yield? And this is an experiment that I was fortunate enough to be able to set up when I first went to Illinois in 1999. It's called free air carbon dioxide enrichment. And basically in these rings, we release carbon dioxide according to wind speed and direction, so that within that ring, the CO2 concentration is continually elevated from planting the crop right the way through to dry down of the crop. So we're elevating it to what we think the level will be in 2050, and the only thing we're altering is carbon dioxide. The rest of the environment is unaltered. So what do we see in these experiments? Well, with soybean, we see about a 16% increase in yield using modern cultivars. In rice, about 12%, and in wheat, about 15%. So when we measure photosynthesis, it's being boosted in these experiments. So we know that if we can boost photosynthesis, we will get more yield. Now, I don't want this to be interpreted as atmospheric change is good for our crops, because that is not the case. And in fact, I was talking to John Pickett about this after his talk yesterday. What we see actually is we're raising CO2 by about 40%, but pest damage increases 100%. So this is the corn rootworm, Western corn root variant. It comes into the soybean at the end of the season, feeds on the soybean, then it lays its eggs. In these elevated CO2 conditions, it lays twice as many eggs. It seems to be able to feed more effectively on soybean than it can under ambient conditions. And colleagues of mine have also shown that this is true of the Japanese beetle. It lives longer under elevated, feeding on soybean under elevated CO2, and produces twice as many young. And it's true of the soybean aphid as well. So quite different feeding yields responding. In separate work, we've shown that increased vapor pressure deficit, which comes along with climate change, is lowering the ceiling on potential yields under rain-fed conditions. And we've recently shown statistically that ozone, tropospheric ozone, which is another greenhouse gas rising with CO2, today costs about 8% of US maize yields, about 4% of US soybean yields. So billions of dollars in lost yield. So what I want to say is that waiting for CO2 to rise is not a solution to raising crop yields. But it does tell us that if we could boost photosynthesis in some way we would get more yield. And indeed, if we go back to this equation, what you can see there is that the conversion efficiency, and this isn't measuring photosynthesis, this is measuring dry matter per unit of intercepted radiation, goes up, and that results in a significant increase in yield. We do see a decline in harvest index, so it does suggest that there is some improvement in partitioning efficiency, sink strength that could be undertaken here as well. So breeders have not managed to improve photosynthetic efficiency to any large extent. Why do we think we could do it now? Well, first of all, photosynthesis is probably the best known of every plant process, of all plant processes. We know every step in the process, we know the genes controlling that, we know the variation there. We know it in so much detail that we can basically model it, which we can now do with high-performance computing. And crop transformation is becoming increasingly routine, even in the public domain. So all of those factors make this a possibility. Well, my group have written several papers on this potential, and one day I got a call from the Gates Foundation saying, can you come out to Seattle with some colleagues and tell us about this? And basically, at the end of the day, they said, well, how about we put our money where your mouth is, and you show us that this can really be done. And so I've said photosynthesis is the final frontier to my students one too many times. And one morning when I came into work, this was stuck on my door. And I walked past and I thought, oh, they're just trying to tell me that I'm first generation Star Trek. But then I realized, no, there's something different about that picture. And then I realized all these faces on there. And these are my co-conspirators, Martin Parry from Rothamsted, now Lancaster, Don Ort from USDA-ARS, Murray Badger from the Australian National University, and Christine Rains from the University of Essex. So basically, what I want to tell you about is this RIPE project, which I was lucky enough to direct starting in 2013. And this diagram really shows you our philosophy, that it's basically, we're using computer models to predict, to simulate photosynthesis in silico, then optimize it and say, well, where should changes be made to make the process more efficient, more resource use efficient, as well as more productive? And then that's a guide of the thousands of transformations we could make, what may be our best bets. Transform those. We're doing that in tobacco, which sounds odd, but we're doing that because it is something that can be transformed very rapidly. But unlike Arabidopsis, it is a crop. So we can take it to the field, it'll form a closed canopy, and it's our first test of concept. If that works, then we go on to the crops that the Gates Foundation really care about, rice, cassava, and cowpea. And in fact, TJ Higgins is here, and we're very much hoping that one of the things we found works in tobacco. Through TJ, we can now possibly test that in cowpea. So we've really honed in on 7 different ways we could do this. Importing algal mechanisms for concentrating CO2 in the photosynthetic apparatus, improving photorespiration, i.e. the energy needed for photorespiration, improving mesophyll conductance, i.e. movement of CO2 to rubisco, the enzyme which catalyzes the carboxylation reaction, identifying better rubiscos from other plants that we could move into our crop plants, improving regeneration of RUBP, that is the molecule which accepts carbon dioxide, optimizing canopies, i.e. sort of redesigning the canopy so that light distribution is more effective. And finally, relaxing photoprotection. And I'll explain what relaxing photoprotection means in a bit more detail, because so far that has been one of our most successful pipelines. Now, of course, the Gates Foundation's questions were, well, you know, we get lots of people coming to us saying they've found a gene that makes Arabidopsis twice the size. People put it in a crop plant and it doesn't do anything. So how are you going to do better than that? And how are you going to convince us that when somebody says they found this in a greenhouse in Canberra, it's better than what somebody in Rothamsted has found in their controlled environment? So the solution to that was to put all of these into one genotype of tobacco and then test that tobacco in field trials in Illinois, replicated field trials, Illinois. So that was our filter of all of these possibilities. And what comes out of those field trials as promising should then move into the crops that the foundation care about. So 4 of those have actually delivered in the greenhouse and are now starting to go into field trials. And what you can see above is us planting this year's field trials. So I want to tell you in detail about a couple of these pipelines. Increasing efficiency in the RUBP production line. So this is the regeneration of the acceptor in the Calvin cycle. Now, the way we've gone about this is a systems approach. And of course, the systems approach was first developed for manufacturing pipelines. You have 1,000 workers on a production line. Where do you put those workers to maximize your output? So if you use systems analysis, what this might tell you is, well, our bottleneck is making the seats. So if we put more workers on the seats, we move our bottleneck somewhere else. And of course, there are millions of possible permutations of this. But high-performance computing can work through these permutations very rapidly. They can do it literally in seconds today for a car production line. Photosynthesis, shown here in this diagram, is a bit more complicated. And there we worked out that there are several billion permutations you could have to maximizing this. Anyway, we, after Xingwan Zhu, when he was my graduate student, spent 4 years working on this, basically representing every step as a differential equation, nonlinear, and then producing that system of differential equations, and then because there's no analytic solution, numerically integrating it. And that's where we got stuck, that it forms a stiff system of equations which are very difficult to numerically integrate. Now, what I want to tell you now is that don't let anybody tell you that crop science is not rocket science, because I can prove that it is. Because the solution came from one of our computer scientists who was working with NASA on balancing propulsion of rockets. And he said, I have exactly the same mathematical problem, and here's the solution. And so we used his algorithms and it worked. And that's why his name appears on our papers. And so we got our system working, we could optimize it, and it said, this enzyme, SBPase, is hugely underinvested. You need 10 times the amount that's there. And yet it's a very small amount of protein. So then, of course, when you've optimized that, you move the bottleneck somewhere else. And that next step was FBP outlase. So to cut a long story short, my colleague, Christine Rains, put these in sequence into tobacco. And so next to the wild type, you can see a representative plant of the SBPase upregulation, then adding in the next one, and so on. And so here are 3, and the third one is more productive. And in greenhouse and field trials, we've shown those to be more productive. Of course, one of the things you might ask is, well, why didn't nature do that? And actually, what we found was if you grow those plants at 220 parts per million of CO2, which is the average of our atmosphere over the last 25 million years, it doesn't do anything. But if you do it at today's CO2 concentration of 400, now it does something. And so what we think has happened is, as CO2 has risen, we've unbalanced the system. What nature optimized over 25 million years has become unbalanced, and this is rebalancing it. My colleague Don Ort has been working on putting in bacterial glycolate metabolism into tobacco to reduce the energy cost of photorespiration. Now, this has been done before, but what Don has done differently is to shut down most of the native pathway. And here you can see an early product of that, where he's clearly under greenhouse conditions getting more productivity. So what I now want to move, sorry, I'm going in the wrong direction. And I want to move on to is photoprotection. And I've called this making the most of the shade because it's about making leaves more efficient under shade conditions. So a leaf at the top of a crop canopy in full sunlight is typically receiving more sunlight than it can use. It absorbs that sunlight, but it can't use it in photosynthesis. It doesn't have enough capacity. So what it does is it induces processes which convert that excess energy into heat. If it didn't do that, what we'd have would be triplet state chlorophyll, which is highly oxidizing, which would begin to auto-oxidize the whole photosynthetic apparatus. So it's called photoprotection. But of course, when the leaf goes into the shade, and it is now limited by the amount of sunlight it has, it could use all of that in photosynthesis, it still carries on dissipating some of that energy as heat. And it can take 30 minutes for this to fully relax. Under some conditions, it can take literally hours until it actually gets to this situation where it's using most of that limiting light in photosynthesis. And our modern crop canopies, for example, soybean, maize, have a leaf area index of about 6 or 7. That means you've got a huge amount of the canopy which is under shade conditions some of the time. So more than 10 years ago, we modeled what this might cost, because at the level of a chloroplast, as the sun crosses the sky on a clear sky day, those lower leaves are going in and out of shade. So as the sun moves across the sky, you can see in this layer 2, 1 second it's in full sunlight, the next second it's in 1/10 of that. And as you go down the layers, this shading becomes more and more frequent. So if you combine this effect together, it says that you could be losing about 20% of your potential productivity. So what we've been looking at is, well, can we speed that up? And so we've been upregulating the genes which induce this protection and remove this protection. And because we can identify plants which are doing this more rapidly through modulated fluorescence, we can quickly pick out the best of our transgenic events. So we can do that by imaging, as you can see in this picture, and then we can grow these under fluctuating light conditions. And what we can see here is 3 events which, under those fluctuating light conditions, are clearly doing better than the wild type from which they're derived. And we are seeing, on average, about the best events, 20% improvement in productivity, on average about 15%. So in total dry weight of those plants, they're taller, they have more leaf area as well. But of course, what we really care about is what happens in the field. And our hypothesis is that our ripe plants should be doing better in the field than our wild-type plants. The ripe plant here is Vanacromdike, and the wild-type is Kasia Burshak, who are the two postdocs who've really done most of this work. And what we see in the field is these were planted exactly the same day in these replicated plots. On the left, you can see the wild type. On the right, the transform plant, which is clearly considerably more productive. So one, of course, other question we come up with is, well, what about C4 photosynthesis? And I should say, the Gates Foundation have a separate project from ours called C4 Rice, where a group of researchers are putting the C4 process into rice. But we, of course, maize, sugar bean, and sorghum are some of our most productive crops. Can we improve photosynthesis in those? And so DOE put out a challenge through ARPA-E a while ago, high-risk projects. So we said, well, we'll give you a high-risk project, improving photosynthesis in sugarcane and sorghum. And so, again, what we've done is to similarly model C4 photosynthesis, which was more of a task, and Yu Wang, who's a postdoc in my group, did this work in a PhD in collaboration with Jingwan Zhu. Again, we got the C4 process working in a similar way. We were able to optimize it and then say, okay, where are the limitations? And it comes up with Rubisco, which is not surprising because that's such a huge protein and nitrogen cost, but also pyruvate pi dikinase, which is a much smaller amount of nitrogen in the plant. So we upregulated that in sugarcane and sorghum. These are results for sugarcane. This is just showing, okay, we showed the gene, the one we put in comes from miscanthus, using the miscanthus promoter and also enhancer elements. Put that into sugarcane, which is quite closely related to miscanthus. And so we found from PCR it was expressing. We then looked at enzyme activity, and most of those events we'd significantly boosted enzyme activity. Now, when we looked at light-saturating photosynthesis, this was when we started dancing around the room because sugarcane has remarkably high rates. This is 45 micromoles per meter squared per second. You never see that with wheat, soybean, or rice, unfortunately. But what we were able to show is that in those 7 events, all of them were showing significantly higher rates. And you might say, again, why hasn't evolution done this? And this picture of assimilation versus CO2 perhaps tells you why, because that initial slope is limited by the amount of PEP carboxylase. Under pre-industrial CO2 conditions, you would almost always be on that initial slope. But what Rubisco and pyruvate pi dikinase are doing is regenerating phosphoenolpyruvate for PEP carboxylase. When you go to higher CO2, you're now on this plateau, and that is governed by pyruvate pi dikinase and Rubisco. And what you can see is in the transform lines, we've raised that plateau. And again, it's something that gives you a benefit under today's CO2 concentration. Our colleague in this work, Freddie Altpeter in Florida, has then tested those lines in replicated trials in the field, and 6 out of 7 of those events are showing higher productivity, averaging about 6 tons dry matter per hectare more. And Syngenta are now independently testing these events. So, of course, one of the exciting things for me, having worked on low temperature tolerance of C4 photosynthesis for 40 years, is that we'd also predicted from our modeling that this enzyme was critical for low temperature photosynthesis. And what we see when we lower the temperature to 11 degrees, which is very chilling for sugarcane, is that photosynthesis in these events is about 67% higher. So we're certainly pursuing that. So I hope I've kind of convinced you that we can now improve photosynthesis in our major crops, and there will be benefits in terms of productivity and sustainability because we're investing resources in photosynthesis more efficiently. If you want to know more about these projects, there are two websites, ripe.illinois.edu and petros.illinois.edu. And finally, I should say that, as would be obvious, that this work is the result of many current and former graduate students and postdocs and faculty colleagues. And this is an incomplete list of the people that actually did the work that I've presented to you. So thank you.
Thank you so much for that, Steve. It's and it's very encouraging just to see in the field that you know you don't even need statistics to convince yourself things are happening there. The equation that you talked about right at the very beginning from Rothamsted included the partitioning parameter as well. Now, do you think with these higher biomass events that you are beginning to generate, that the partitioning and therefore the harvest index will need to be worked on separately, or do you think it will actually compensate and therefore deliver more grain filling?
Yeah, I think that's probably going to be very crop specific. You know, from our experience of soybean, we think that, you know, as you boost the productivity, photosynthetic efficiency, that the work on harvest index and partitioning will be necessary. I guess Longping Yuan was pretty convinced with the best rice cultivars that they're waiting for more source. And this may, you know, what you're saying may be the case with wheat as well. Under elevated CO2, harvest index did drop a little bit. I think with crops which have had less attention, this may be far more important, that you're going to need to improve harvest index along with photosynthesis.
Beautiful talk, Steve. I'm not my expertise is not photosynthesis, so maybe this question is a bit simple-minded, but it has to do with the linkages between harvest index and the conversion efficiency. As you mentioned, the harvest indices we see in crops is fairly close to the theoretical maximum, about 0.65. And I think of, you know, efficiency in harvest indexes involving vascular biology, efficiently moving the photosynthate from the source leaf to the seed. And I was wondering, and of course, if that's not moved efficiently and you have photosynthate building up, you could have feedback inhibition on conversion. Is that possibly happening, or is it already so efficient that you don't have that happening? I'm just wondering if that's another way to impact conversion efficiency.
I think that can certainly be the case under some conditions. You can show, if you limit sink size, that you are going to get feedback inhibition of photosynthesis. So again, I think, you know, as I was saying in answer to the other question, I think it's going to be very crop and cultivar specific, that, you know, this is going to be most effective where breeders have really done a great job of improving sink size. And where that hasn't happened, there'll almost certainly be feedback inhibition of photosynthesis.
Thanks.
Maybe an extremely insane question. If humans have now all the background technology to engineer chloroplasts into animals, how would the animals would take it, and what would be required, and would it have an impact on the global carbon budget?
it certainly will have an impact on the global carbon budget. You know, and I guess we all hope that's going to be the case. If these projections of food demand are correct, then I think there's no way around that. Whether we do this through photosynthesis, improved agronomy, whatever, we are going to affect the global carbon budget. There could be some minor benefits for global change from this. We know that, you know, as crops become more productive, and, you know, this has been demonstrated in the Midwest with corn yields in particular, more carbon is actually put back into the soil. So we do have that benefit from more productivity. But I'm afraid if food demand continues to increase, then I don't think we have any, if people are going to be fed, we don't have any choice but to affect the carbon budget.
I think I missed about into animals. Like if you have the photosynthetic animals, the animals which have now chloroplast machinery in them.
Oh, yeah. Okay, animals. Yeah, I mean, of course, this is something that can be done with genetic engineering, but the key, the reason why plants have leaves is they're dorsiventrally flattened to make them highly efficient at capturing CO2 from the atmosphere. Animals do not have that leaf area. So if you do the sums, the amount of energy that a green cow could convert into carbohydrate is less than 1% of its daily requirement of calories. Yeah.
Thank you.
So,
Actually, isn't that what you did with your postdocs? Because they look green. So. Okay, I have another quick question, which is, given that it's theoretical, how do you know what the theoretical maximum is on any of those parameters in your initial equation?
oh, okay. That is, you know, we know every reaction involved. We know, so as soon as light is captured by a chlorophyll molecule, we know the energetics of the transfer of that to the reaction center, the energy costs of electron transport, and then the energy costs of carb metabolism through to carbohydrate synthesis. So those are exact numbers. What, of course, you know, we have that visible light conversion of 9.3%. What that doesn't say, for example, is if you eliminated photorespiration, then that number becomes about 12%, for example. So if you made changes in the process, that's another way of actually doing it. But if you look at it, the transfer of excitation energy after photon capture through to production of NADPH and ATP is, most plants can do that almost at maximum efficiency. The inefficiencies then come downstream of that. And the bulk of those are due to the fact that there just isn't enough capacity in carbon metabolism, particularly at the point of rubisco. But rubisco is already half of the soluble protein. So upping that is really not much of a practicality, unfortunately. So a lot of these modifications are, how do we deliver more CO2 to rubisco to make it more efficient? Can we put in better rubiscos? And, you know, can we just keep the whole system operating at maximum efficiency around that? So.
Well, we've certainly followed on a tradition